Hello friends, today we will learn. It's a second video of transgenic plants. We'll discuss more in this video. So from now we'll discuss about the environmental impact of BT crops. BT means Bacillus thuringiensis crops. The most serious impact of BT crops on the environment is the build-up of resistance in the pest population. In 1999, another issue was brought to light about BT crop. It was reported that the pollen from BT maize might be toxic to the larva of monarch butterfly. This generated considerable opposition to BT crops by the public since monarch butterfly is one of the most colorful natives in USA. It was later proved that the fears about the impact of BT crops on the monarch butterfly were without the required scientific evidence. The lesson learned from the monarch butterfly episode is that the risk of genetically modified crops should be thoroughly assessed before they are reported. Usage of BT. The usage BT is commonly used for the transgenic crop with a cry gene. Example BT cotton. In same way, cry proteins are also referred to as BT proteins. It may also be stated here that the authors use four different names for the same group of proteins: delta endotoxin, insecticidal crystal protein, in short ICP. Cry and now BT. Now let's know about resistance genes from other microorganisms. The BT toxin genes from Bacillus thuringiensis have been described in preceding page. Now we will discuss. There are certain other insect resistant genes from other microorganisms. First is cholesterol oxidase. Second is isopentenyl transferase gene. First is cholesterol oxidase of streptomycin culture filter was found to be toxic to ball weevil larvae. Cholesterol oxidase gene has been introduced into tobacco to develop a transgenic plant. Isopentenyl transferase gene from Agrobacterium typhuscens has been introduced into tobacco and tomato. This gene codes for an important enzyme in the synthesis of cytokinin. The transgenic plants with this gene were found to reduce the leaf consumption by tobacco hornworm and decrease the survival of peach potato aphid. Now let's know about the resistant genes from higher plants. Certain genes from higher plants were also found to result in the synthesis of products possessing insecticidal activity. Some authors regard them as non-BT insecticidal proteins. Now we will discuss about proteinase inhibitors. Protease. Proteinase inhibitors are the proteins that inhibit the activity of proteinase enzymes. Certain plants naturally produce proteinase inhibitors to provide defense against herbivorous insects. This is possible since the inhibitors were, when ingested by insects, interfere with the digestive enzymes of the insect. This results in the nutrient deprivation, causing death of the insects. It is possible to control insects by introducing proteinase inhibitor genes into crop plants that normally do not produce these proteins. Cowpea. Let's know about cowpea trypsin inhibitor gene. It was observed that the wild species of cowpea plants growing in Africa were resistant to attack by a wide range of insects. Research findings revealed that the insecticidal protein was a trypsin inhibitor that was capable of destroying insects belonging to the orders Lepidoptera, example Heliothrix virexcans, Orthoptera, example is Locusta migratoria. and Coleoptera. Example is Anthonus gradis. Cow PE trypsin inhibitor, in short CPTI, has no effect on mammalian trypsins, hence it is non-toxic to mammals. CPTI gene was introduced into tobacco, potato and oilseeds rape for developing transgenic plants. Survival of insects and damage to plants were much lower in plants possessing CPTI gene. Advantages of proteinase inhibitors. Many insects not controlled by Bt can be effectively controlled. Use of proteinase gene along with Bt gene will help to overcome Bt resistant development in plants. Now let's know about the limitations of proteinase inhibitors. Unlike Bt toxin, high levels of proteinase inhibitors are required to kill insects. It is necessary that the expression of proteinase inhibitors should be very low in the plant parts consumed by humans, while the expression should be high in the parts of plants utilized by insects. Now let's know about alpha amylase inhibitors. The insect larvae secrete a gut enzyme alpha amylase to digest starch. By blocking the activity of this enzyme by alpha amylase inhibitor, the larva can be starved and killed. Alpha amylase inhibitor gene isolated from bean has been successfully transferred and expressed in tobacco. It provides resistance against Coleoptera. Example: Jabrots fasciatus. Now let's know about lectins. Lectins are plant glycoproteins and they provide resistance to insects by acting as toxins. The lectin gene from snowdrop has been transferred and expressed in potato and tomato. The major limitations of lectin are that it acts only against piercing and shocking insect and high doses are required. Now let's know about the resistant genes from animals. Proteinase inhibitor genes from mammals have also been transferred and expressed in plants to provide resistance against insects. Although the success in this direction is very limited, bovine pancreatic trypsin inhibitor, in short BPTI, and alpha 1 antitrypsin genes appear to be promising to offer insect resistance to transgenic plants. Now let's know about insect resistance through copy nature strategy. Some of the limitations experienced by transferring the insecticidal genes and developing transgenic plants have prompted science to look for better alternatives. The copy nature strategy was introduced in 1993 with the objective of insect pest control, which is relatively sustainable and environmentally friendly. The copy nature strategy for the development of insect resistant transgenic plants. Now we will discuss. First stage is identification of leads. The first step in copy nature strategy is to identify the plants from world over that are naturally resistant to insect damage. Second is isolation and purification of proteins. The protein with insecticidal properties is isolated and purified. The sequence of the protein is determined and the gene responsible for its production identified. Third stage is bioassay of isolated protein. The activity of the protein against the target's insects is determined by performing a bioassay in the laboratory. Testing for toxicity in mammals. It is absolutely necessary to test the toxicity of the protein against mammals, particularly humans. If the protein is found to have any adverse effect, the copy nature strategy should be discontinued. Next point, fifth point is gene transfer. By the conventional techniques of genetic engineering, the isolated gene corresponding to the protein toxin is introduced into the crop plants. Now let's know about the selection of transgenic plants. After the gene transfer, the transgenic plants developed should be tested for the inheritance and appropriate expression of transgenes. The efficiency of insecticidal protein to destroy insects is also evaluated. Now let's know about the evaluation for biosafety. Field trials have been Field trials have to be conducted to evaluate the crop yield, damage to insects, influence on the environment with respect to the transgenic plant, the copy nature strategy through time, though time consuming and many a time unsuccessful, takes into account the complex interplay in biological communities and physical environment. Virus resistance. Now let's know about the virus resistance. Virus infections of crops may result in retarded cell division, plasia, excessive cell division and cell death. The overall effects of virus infections are growth retardation, lower product yield and sometimes complete crop failure. The chemical methods used to control various plant pathogens will be ineffective with respect to plant viruses since the viruses are intracellular obligate parasites. There are, however, certain safe agricultural practices to control or reduce viral infections to plants. Use of seeds that are virus free. Use of seeds that are virus free. Next point is control insects that spread plant viruses. Control weeds that serve as alternate host for viruses. Use cultivars that possess virus resistant. It is possible to immunize plants against viral damages by expressing viral proteins in the plant cells. With the advances made in genetic engineering, it has become a reality to develop transgenic plants with virus resistance. This is mostly done by employing virus encoded genes, virus coat proteins, movement proteins, transmission proteins, satellite RNA, antisense RNAs and ribosomes. In recent years, some attempts are also made to provide virus resistance to plants by using animal genes. Let's know about this animal genes. Virus coat proteins. First is virus coat protein. This is the virus resistant. Virus coat protein. Let's discuss. The virus coat protein mediated approach is the most successful one to provide virus resistance to plants. It was in 1986 transgenic tobacco plants expressing tobacco mosaic virus coat protein gene were first developed. These plants exhibit high levels of resistance to TMV. TMV means tobacco mosaic virus. Excited by this remarkable success, scientists have worked with many more viruses, around 30 or so, and developed crops with virus coat protein mediated protection. The transgenic plant providing coat protein mediated resistance to viruses are rice, potato, wheat, tobacco, peanut, sugar beet, alfalfa, etc. Thank you. We'll next discuss in next video.